still people coming in, but that will always be the case, even if we do it physically. Welcome to everyone. Welcome to the speakers, but especially welcome to all of you. We were very happy to see in the participants list and the list of registrations that you come from all over the world. We're very happy about that. My name is Diederik Sprangers. I am the scientific coordinator of ENSSER, the organization who organizes this roundtable about Corona. First of all, I should apologize for the noise of my computer. You may hear a high-pitched sound of the ventilator of my very old computer, which is trying to keep cool, as I am. ENSSER, what is ENSSER? ENSSER is a European network of independent critical scientists, and our goal is to advance science for the protection of the environment, biodiversity, and human health, particularly from adverse impacts of modern technologies. So we've particularly done a lot of work on genetic engineering, but that's not the only thing we're active in, by far not. Please take a look at our website to see what we do. We have been deliberating within our organization for a long time this year whether we should do anything, say anything in public about the current Corona pandemic. We wanted to, many of our members wanted to keenly, but we had long and good discussions. And from the science point of view, and I think from almost any other point of view, it is still a rather confusing situation. But from science particularly, it is even now, after nearly a year since it started, it's very hard to give any answers to any questions on Corona, on COVID-19, I should say. But at length, we decided we had something to say, and 3 of our members are doing so to you this afternoon. In the first place, Dr. Michael Antoniou from King's College London School of Medicine. Then Professor Ignacio Chapela from the University of California, Berkeley, whom we're very honored to have because it is 4:30 in the morning in California, so he's sacrificing part of his sleep for us. And the third speaker is Professor Giuseppe Longo of CNRS in Paris. And then in the second half of the program, later this afternoon after the break, we'll have a different moderator, not myself, but Dr. Angelika Hilbeck of the Swiss Federal Institute of Technology, ETH, in Zurich. Dr. Hilbeck is also one of the founding members and a board member of ENSSER. ENSSER was founded 11 years ago, 2009. So we waited long because we really want to give a measured and well-thought intervention in this confusing situation. That is important to us. Critical science is really being careful about what you say, but not keeping your mouth shut if you want to say something. A few practical things. In the first half of the program, the 3 talks until the break at 3:30, you can ask questions for clarification only, please. And you can only do so in writing, not in speaking yet. So please use the Q&A button at the center bottom of your Zoom window. If you press it, you can write a question to that particular speaker. But please ask questions only for clarification. There will only be about 5 minutes after each talk for these questions. So we're not taking any time for comments or for discussion now. Please save those remarks or questions for the discussion in the second half of the program. Then after the break, you will be able to ask written questions or make written comments and oral questions or comments. And to do that, you need to press the raise hand icon at the bottom center of your Zoom window. then we see your hand raised in the participants list, and the moderator, Dr. Hillbeck, will give you the word, will give you permission to speak, will give you the chance to speak. If you want to say something, or if you're writing a question or a comment, please mention your name and affiliation. Your name may already be in the name that you registered with, but some people sign with a pseudonym or a different word. So if that's not your name, then please state your name and your affiliation, the organization you work for, or if you're speaking as a private citizen, just say, I'm speaking as a private citizen. We're not accepting any anonymous contributions. We'd like to see who is speaking to us and be in open contact with you. Now, COVID-19 is a controversial topic, so I could imagine there are some people who really who have a good reason to remain anonymous and would still want to address us. If that would be the case, please do not do so in this public event, but address us privately, send us a private email to our office email address, or use the contact form on our website. Our office email address is simply office@ensser.org. I'll be very strict on the time. I'm nearly done. Each of the speakers has about half an hour, including questions for clarification only. And then at 3:30, we'll have a break. But just before that break, I'll hand over to the other moderator, Dr. Hillbeck. I think that was all I should say now. And I'd like to hand over to the first speaker, Dr. Michael Antoniou. He's a molecular geneticist, as I said, from King's College London School of Medicine. And he will speak on the virus, SARS-CoV-2. Is it a natural original, or is it a laboratory creation? And does that matter? Michael, the screen is yours.
Thank you very much, Diederik. Just first of all, I wish to thank ENSSER for setting this up, actually holding this event on such a vastly important topic. And I should declare that I have been a member of ENSSER from its first year, and it's been an honor to be so. So let me share my screen with my PowerPoint presentation. So, as Diederik said, the theme of my presentation is the SARS-CoV-2 virus, the cause of COVID-19. Is it a natural, original, or laboratory creation? Should say natural. Does it matter? My presentation, there have been many commentaries, papers published on the topic of the origins of SARS-CoV-2. There's no way I can cover all of them. I can't claim to have read all the papers and opinions on this. What I plan to present here to you are some key observations on whether SARS-CoV-2 is of a natural origin, or in other words, a zoonotic, an animal origin that has then mutated and been selected in human populations, or whether it has been intentionally created and accidentally escaped from a laboratory. And why does this matter? Because today, one of the themes, we're addressing is science in the spotlight. So what is the role of science in this area? Before I go on to present some evidence, I would just like to review with you very briefly the known mechanism of coronavirus infection, which is illustrated on this slide here. Basically, coronavirus, illustrated here on the left, the envelope is coated by what is known as the spike protein, among other things. And the spike protein consists of, that protrudes from the virus envelope, consists of two domains, what's known as an S2 domain, yeah, and also the S1 domain. Now, the S1 domain contains what is known as the receptor binding domain, the RBD here. So, and as that name implies, the S1 domain of the spike protein protruding from the coronavirus, in this case SARS-CoV-2, is what binds to receptors on the cell surface of cells, in this case ACE2. So coronaviruses bind to ACE2 receptors on the cell surface via the spike protein region receptor binding domain. But in order for the virus to actually infect the cell, to enter the cell and infect it, what next needs to happen is that the spike protein needs to be cleaved between the S1 and S2 domains. So a protein cleaving enzyme, in this illustration that I have here for you, this protein cleaving enzyme, for illustrated purposes, is TMPRSS2, which cuts between the spike protein S1 and S2 regions, as illustrated on the right-hand side here. Upon cutting between these two domains, the protein cleaving enzyme, the S2 region of the spike protein is able to mediate the entry of the virus into the cell through one or more mechanisms. We don't have to worry about that at the moment. Only the crucial points I want you to take home, to take into account from this slide, is that there is the spike protein is key, at least it's thought, is the key to the infectious process, and there needs to be a cleavage between the S1 and S2 domains of the spike protein in order for the infection to properly take place. So we need a protein cleavage enzyme, which is provided by the cell. So these two things, we need, as we go forward, please bear these in mind, because I'll be referring to this mechanism as we go along. Now, when the COVID-19 pandemic broke out, I have to admit to you, I had no intention of getting involved in any of the debate on this. But then as things got published and I read them, I began to get a bit concerned about the information about the SARS-CoV-2. The first paper that was brought to my attention was this one published by Anderson and colleagues. And it was actually a correspondence, just the letter to Nature Medicine, not a fully blown paper, entitled The Proximal Origin of SARS-CoV-2. And this is a group of scientists based at Stanford University in California. What they basically showed in this paper was, they did a sequence analysis of RNA genomes of coronaviruses. The genomes, the genetic material of coronaviruses is RNA. So they did a lot of comparisons of the RNA genomes of different coronaviruses. And then following on from that, based on that, they used a computer prediction program in order to determine what they believe would be an ideal coronavirus sequence for human infectivity, especially around this receptor binding domain that I mentioned before of the spike protein. And their conclusion was that actually the prediction of the computer prediction of an ideal coronavirus for human infectivity did not fit the computer model of, or I should say, the SARS-CoV-2 did not fit the computer model of an ideal coronavirus for human infectivity. And based on that, they presented this as evidence. And in their view, and I think this is where they went well beyond what the evidence allows you to do, they said that SARS-CoV-2, their evidence provided almost definitive evidence that SARS-CoV-2 cannot be created in the laboratory. Now I thought, now this can't be right, because being a molecular biologist, I've been manipulating genes and know of gene manipulation, of technologies of all sorts. I felt that what these authors ignored was, first of all, they put blind faith in their computer predictions, bearing in mind that computers could only predict, they cannot provide with definitive evidence. You have to do experimental work to confirm computer predictions. And these authors ignore laboratory methods for creating and selecting coronaviruses for human infectivity. For example, iterative evolutionary selection. I tried to publish a letter. I submitted matters arising to the journal Nature Medicine to try to point out these deficiencies in this paper, but was rejected by the editors. But you can read this letter via this web link. So this type of thing got me very suspicious as to, maybe people are not being completely forthright about the nature of SARS-CoV-2. And this included claims by some people that if SARS-CoV-2 was intentionally laboratory manipulated and accidentally escaped from a laboratory, the genetic manipulation would leave signatures behind in the genome of the SARS-CoV-2. Now, again, I've been manipulating genes for decades, and I can guarantee I've been doing that without leaving any signature behind that I actually even manipulated those genes. So I knew for a start that the SARS-CoV-2 genome could be manipulated and in such a way that you wouldn't know that you would have a manipulated, say, a bat virus and converted it into a human infectious virus. So that got me kind of thinking, because I thought that the science, we were not being completely loyal to the science. And of course, the next paper that came out even, I think, before the Anderson paper, there's, of course, if you want to postulate that SARS-CoV-2 had a natural origin, and which then mutated and got selected for human infectivity, you need to propose what is known as an animal proximal origin of SARS-CoV-2. And this paper from Chinese scientists led by Zhengli Shi, who heads the Wuhan Institute of Virology, they basically published this paper in Nature back in January, that a bat strain of coronavirus, which they designated as RATG13, they proposed it as a proximal origin virus for SARS-CoV-2, because they say overall the SARS-CoV-2 and the RATG13 had a 96% sequence identity, they were the same in 96% of cases, with differences, say, across the receptor binding domain of the viruses. However, this RATG13, analyzed by other commentators, they found a number of anomalies about RATG13, which questions its possible proximal origin status. First of all, RATG13 was supposedly originally isolated from bats in Yunnan province, which is several thousand miles away from Wuhan. And a partial sequence of RATG13 was posted in 2013. So the first question that arises is, why wait so long to report on this bat coronavirus, RATG13, which clearly, based on its sequence, has the potential to mutate and cross to humans? Surely it would have been a duty of the scientists responsible to make this sequence publicly known and warn the world as a warning that there is this virus out there that could possibly mutate and be selected for human infectivity. Secondly, when the sequences of RATG13 and SARS-CoV-2 were analyzed, they did not diverge in accordance with principle of evolutionary genetics, especially in the spike protein gene region. And one of the genes of SARS-CoV-2 was the E protein gene, was actually not only similar to RATG13, but actually identical in sequence to two other bat viruses that were reported by Chinese scientists, ZC45 and ZXC21, which were published in 2018. Now, the key thing here is that the E protein is the E protein gene region mutates quite rapidly in coronaviruses because mutations in this protein are well tolerated. So mutations, base sequence changes accumulate very rapidly in the E protein gene. But why do we have here SARS-CoV-2 having an identical sequence to the ZC45 or ZXC21E proteins when they're supposed to be so many years apart in where they when they arose. So it raised the question as to actually whether R8-TG13 actually really exists or not. And today there are no isolates available. And as far as we're aware, it's just a sequence in someone's computer. And you can read a commentary on this website here. The next paper, which again kind of makes the origin of SARS-CoV-2 difficult to comprehend, is this paper published by Alina Chan, based at MIT in the United States. SARS-CoV-2 is well adapted to humans. What does this mean for re-emergence? What Alina Chan presents in this paper is quite a lot of very, very well conducted genetic sequence comparative information. But for me, this is the key observation is summarized in this one figure from her paper, which is a comparison of the genetic progression of SARS-CoV-2 and SARS. SARS-CoV is the SARS of the 2003 epidemic that arose in China. And she compared the genetic progression or changes in the genetic sequence of the 2003 virus, SARS virus, and the SARS-CoV-2. And what you see on the left is what you would call what you see as a classical progression of genetic mutation, selection, adaptation of a virus as it goes from an animal to a human. In that, over the first few months, there is marked progression. Very many mutations begin to accumulate. As it goes from round after round of human infection, you get very large numbers of genetic mutations accumulating until towards the end of, you see far fewer. Once the virus is basically adapted to a human host, you see far fewer genetic alterations beginning to happen. Why is that? Because any further mutations are likely to make the virus less adapted to humans rather than more adapted to humans. So this is the classical progression of adaptation that you would see genetically in a virus to human, and an animal to human viral transmission. On the right here, you see the genetics of SARS-CoV-2 from December to March, when again, the virus was moving through human populations. And what you actually see is actually very little change in the base sequence of the SARS-CoV-2. It's as if even by December last year, the SARS-CoV-2 appeared already adapted to humans, highly adapted to humans, and didn't need to go any further, was no further adaptation needed. And so there's one or two ways in which you can explain this. One is either that the SARS-CoV-2 had been around in human populations for far longer than is being admitted by, say, the Chinese authorities, or of course, it escaped from a laboratory where it had been adapted in the laboratory for human infectivity, and so it appears in this way and not needed to further adapt. And this adaptation, bear in mind that the adaptation of SARS-CoV in 2003 occurred through, when the numbers of infected individuals were up to around 7,000. Whereas this lack of further adaptation of SARS-CoV-2 is observed even after 1,000 times more rounds of infection, around 800,000 rather than 7,000 rounds of humans being infected. So Alina Chan, in correspondence with people at GM Watch, has said the sudden appearance of highly infectious SARS-CoV-2 presents a major cause for concern that should motivate stronger international efforts to identify the source and prevent near future reemergence. In this context, they also concluded that a lab escape should be considered regardless of how likely or unlikely. In support of Alina Chan's conclusions about SARS-CoV-2 appearing already highly adapted for human infectivity is this paper by Nikolai Petrovsky and colleagues, where they note, notably SARS-CoV-2 spike protein had the highest overall binding energy of human ACE2, greater than all other tested species, including bat, and the postulated source of the virus. This indicates that SARS-CoV-2 is a highly adapted human pathogen, and questions whether the origin was in fact a bat virus. In an interview for the Washington Times in Australia, Nikolai Petrovsky says that the virus's ability to bind human cells far exceeds its ability to infect other animals. And this, plus the fact that no corresponding virus has been found to exist in nature, leads to the possibility that COVID-19 is a human-created virus. It is therefore entirely plausible that the virus was created in the biosecurity facility at the Wuhan Institute of Virology by selection on cells expressing human ACE2 receptors, a laboratory that had been known to be cultivating exotic bat coronaviruses at the time. Now, just to remind you I want to now turn to some of the features of SARS-CoV-2, that for me, genetic features, that for me makes it suspicious that it was a naturally adapted virus. So remember here, just what is said is that the S1 domain, via the S1 domain of the spike protein, SARS-CoV-2 will bind to ACE2 receptors. Then the receptor, there will be cleavage of the spike protein between the S2 and the S1 domains by a protease, and that facilitates entry of the coronavirus into the cells. However, this paper published by Sorensen, Dalgleish, and Susrud questions the necessity of ACE2 receptor for infectivity. They're proposing evidence of ACE2 receptor independent infection by SARS-CoV-2 in a paper entitled "The Evidence Which Suggests That This Is No Naturally Evolved Virus." But basically, just to summarize their key observation in this paper, is that by looking at the sequence of SARS-CoV-2 and its corresponding amino acid sequence, they identified what they called insertions of clusters of amino acids around the receptor binding domain of SARS-CoV-2 spike protein. And this is illustrated on the left. Now, these amino acid insertions give the region around the receptor binding domain an overall positive charge. And these very experienced virologists, they predict that this positive charge, high positive charge around the receptor binding domain, allows binding of the virus and infection of cells that does not possess the ACE2 receptor. And as an example, they illustrate this with the coronavirus causing swine acute diarrhea syndrome, which infects cells through an ACE2 receptor independent pathway. And so they're saying that these clusters, these inserted amino acids that give this positive charge to the region, contribute to an unusual factor of SARS-CoV-2, which is that it's able to infect many cell types and tissue types around the body, unlike usual SARS or coronaviruses. And that their view is because of the number of these inserts, it's extremely unlikely to have occurred by natural mutation and selection. And the second, for me, very unusual feature, suspicious feature, you could say, about the SARS-CoV-2 receptor binding domain region is the presence of what is known as a furin cleavage site, which is unique to the SARS-CoV-2 within its class of coronaviruses. And these two this was the unusual presence of this furin cleavage site. I'll be explaining what that is shortly. But the presence of this furin cleavage site within the spike protein of SARS-CoV-2 was pointed out as being extremely unusual by commentators very early on in this saga, we could say, about the COVID-19 situation. And these are two of the commentaries that I've drawn information from. Firstly, what is furin and why is its presence between the S1 and S2 domains of the spike protein and enhance widespread infectivity. Basically, furin is a protein cleaving enzyme, and it's widely distributed around our body. And that furin, the furin cleavage site within SARS-CoV-2 is inserted between the S1 and S2 domains of coronavirus. And that this would result in widespread infectivity after ACE2 receptor dependent or independent cell association. So basically, SARS-CoV-2 has been pointed out as being unique in its class of coronaviruses, what are known as beta coronaviruses, in having a perfect furin cleavage site between the S1 and S2 spike domains. And that this furin cleavage site, just to show you this table here, I'm not expecting you to read it, but this is a list of all the known beta class coronaviruses, which SARS-CoV-2 is a member. And this compares their sequence between the S1 and S2 domains of the spike protein. And only SARS-CoV-2 has this furin cleavage site at that position. None of the others have. It's almost as if there's an evolutionary selection for not having a furin cleavage site at this position rather than selecting for its adoption. But basically, the presence of the furin cleavage site between the S1 and S2 domains of the spike protein of SARS-CoV-2 is absolutely required for its infectivity, because as this paper by Hoffman and colleagues in Molecular Cell earlier this year showed, that when they deleted the furin cleavage site from SARS-CoV-2, it abrogated or negated its infectivity. And furthermore, there is the underlying genetic sequence that encodes for the furin cleavage site within SARS-CoV-2 has an unusual genetic code or codon sequence. This CGG codon genetic code for part of the furin cleavage site here, this PRRA amino acid cleave recognition site, these are very unusual, very rare, and again a sign that maybe they're unlikely to occur naturally. So in summary, what we have is a perfect furin cleavage site. It is perfectly located between the S1 and S2 spike protein domains. Then it uses a highly unusual coding usage, which can allow its easy detection via this FAL1 restriction enzyme, by this particular DNA cutting enzyme, and raises the possibility that it has been inserted by intentional manipulation. And the other thing I wish to raise as part of our discussion of science in the spotlight in particular is this coronavirus gain of function research, which is the intentional creation of human infectious coronavirus. Scientists in China and in the USA working independently and in collaboration with the USA funding work in China, working on coronavirus gain of function research for over 10 years. And this has included attempts to convert a bat coronavirus into a human pathogen. And these are just two papers. There are many papers on this gain of function research by Chinese and American scientists for coronavirus. One of them is this: a SARS-like cluster of circulating bat coronaviruses shows potential human emergence. Collaboration from the Zhengli Shi laboratory at the Wuhan Institute of Virology and the Ralph Baric lab at the University of North Carolina. And what they showed here was that the fusion of bat and mouse coronaviruses unexpectedly resulted in high human cell infectivity. In another paper, authored by this group of American scientists, entitled Furin Cleavage of the SARS coronavirus spike glycoprotein enhances cell-cell fusion, but does not affect virion entry, what these authors elegantly demonstrated was that actually intentional insertion of the furin cleavage site between the S1 and S2 spike protein domains of coronavirus markedly enhanced what we could call the early stages of infectivity. So in conclusions, what can we say? Is there definitive evidence of a zoonotic animal natural mutation and selection origin of SARS coronavirus? I would say no. is there definitive evidence that SARS-CoV-2 was either propagated and or genetically manipulated in a laboratory and accidentally escaped? I have to say, there is no definitive evidence for this either. But what we do know is this, in my view, that the arguments as to how SARS-CoV-2 arose, either through natural mutation, selection, or laboratory creation, offer alternatives to the same end and do not exclude each other. None of the arguments for natural mutation and selection exclude the natural creation. They only offer alternative pathways to achieve the SARS-CoV-2. However, the SARS-CoV-2 possesses several unique genetic and structural features. And I've only covered a few of them, by the way. There are a number of other unusual genetic and structural features of SARS-CoV-2, which I would say, you know, if there was one unusual feature, you could say, yeah, you could see how possibly it could arise naturally over time. But when you have several, you know, I think the coincidence, it becomes more than a coincidence. And that on balance, the evidence for me, and this is a purely personal opinion, I think the evidence favors a laboratory creation and accidental escape of SARS-CoV-2. I'm prepared to stick my neck out on that based on the current evidence. We also have clear evidence, published evidence that coronavirus gain-of-function research has been going on for many years, and that the knowledge to create SARS-CoV-2 has been there for many years. We've known about how the receptor binding domain interacts with the ACE receptor. We know what's important. We know about how inserting a furin cleavage site could enhance the infectivity. So it's quite easy. To be honest, I could go to the literature, use my genetic engineering skills, and I could create for you SARS-CoV-2. So finally, for points of discussion, science in the spotlight is the theme. Why has gain-of-function research of coronavirus and other pathogenic viruses ever been allowed? And for me, a shameful thing for science at the moment is why are the mainstream journals refusing to publish evidence by scientists providing a laboratory propagation and or manipulation and escape of SARS-CoV-2? And the other final thing is SARS-CoV-2 causes COVID-19, but is it the cause of the pandemic? Thank you very much.
Michael, thank you so much for this talk. You're a model speaker, not only for expressing your thoughts on this difficult subject so carefully and clearly, but also for sticking so well to your time. I see one question from a participant, Helena Paul, from a non-scientist, she says, so I take it she's speaking as a private citizen.
Yes, I know Helena.
Why is all this gain-of-function research carried out? That's on the edge of being clarification and discussion. So very briefly, please answer it.
In many ways, I would like to leave it for discussion later, but overall, it said the way that it's justified is, well, if we know more about how the virus can mutate and gain human infectivity, we can better prepare ourselves for future epidemics or pandemics. I mean, I will come back to this later if we want, because I think that justification holds no water at all.
Right, thank you. There's another question right now from another private citizen, Jelle Schuttenreier. The initial question has not been answered. He says, does it matter whether it is from a lab?
Yeah, again, it's a discussion point, yes, mostly. It does matter because, first of all, for me, it puts the spotlight on science here, because science is at the core of this potentially here, yeah? It's at the core of how has this virus arose and what scientific input there's been in how the apparent pandemic has been handled. So it does matter, or the source does matter, because it shows us whether science has been loyal to science, true to itself, or whether it actually has not been true to itself.
The asker is satisfied. Thanks for asking, Jelle, because this is really what science should be for. There's another question from Sainat Suryanarayanan. US right to know from the United States. Could the speaker clarify his last question? SARS-CoV-2 causes COVID-19, but is it the cause of the pandemic? What did you mean by that, Michael?
Again, it's what has been the way it's been handled. Again, I love this question, and I would like, it will take me some time, and it's a discussion point. If we can save this and bring it back to the end, because I think it's been the way governments have handled this have been highly questioned. So yes, of course, we have a disease and it's caused by this virus, but could this have been handled with better scientific input so that we didn't end up with this pandemic and with people suffering the consequences of lockdown and so on? Yeah. So I'd like to come back to that at the end if possible as well, because there's evidence about whether was the pandemic caused by the virus or by the way it was handled.
Yeah, it depends what you call a pandemic.
Yeah,
of course.
Yeah.
Okay, I'll remind you, or maybe Angelica, I should ask Angelica to remind you of this question in the discussion. Then there's another question from Ole Fergman. He's a member of ENSSER and he's a retired physician, I know that, but he's speaking as a private citizen, I take it. Did the Berger-Sørensen paper show that the amino acid insertions rendered ACE2 unnecessary?
Yeah, well, that is their prediction. They're saying that, of course, it doesn't exclude. Again, what you have is what they call co-receptor. They call it SARS-CoV-2 displays a co-receptor infectivity profile. It can still, of course, through the receptor binding domain on the spike protein, still bind to ACE2 and infect through this pathway. But they're saying that the inserted positively charged amino acids that they've identified, they could allow binding to the cells that do not have any ACE2 receptor, and still you can get the infection cycle taking place through cleavage between S1 and S2 and so forth. So there are two co-receptor pathways rather than one excluding the other.
Thank you. I see one of the participants has raised his hand, but please write your question in the Q&A box if you can. We're not taking any oral questions right now. There is another written question from Hervé Lemeur. He's an NGO counselor, and he says, 1, the ideal sequence for human infectivity is defined only by the scientists and what the scientists know of their programs. 2, you say, I've been manipulating DNA without letting a single signature. You said you could have, I think.
Yes.
I disagree with you. Any genetic modification technique leaves some scars. They may be far away and not under the street light. And 3, you favor to warn the word of dangers. I'm not sure it is necessarily always good. Michael.
Firstly, it's very easy to change a gene sequence without leaving a scar. I mean, the easiest way these days is to just sequence, is to synthesize a region of interest where you, but with the base changes of your desire, and there you have it. A sequence of, say, coronavirus, spike, receptor binding domain, and I've synthesized it. I haven't left any sign of my manipulation there at all, because all I've done is I've changed the just base units in my synthesis. And as I say, I've been doing this sort of thing for many, many years in my studies of various regulation of gene expression studies. So that part, I think, is well known. Any molecular biologist will tell you that they can manipulate without leaving any scar behind. That doesn't mean to say they haven't, by the way. Some commentators have suggested that you can identify some changes in the sequence of SARS-CoV-2 that indicate a possible left behind a kind of signature or an alteration that leaves a signature for manipulation. That may be right or not, but that could arise without any, without the kinds of signatures being there. The other part about the danger, I wasn't quite sure what.
It's three different remarks, really, or questions, isn't it? The last thing says you use the word danger, I suppose. I'm not sure.
About the gain of function.
Yeah.
Yeah, the gain of function is always dangerous, obviously, because you're intentionally trying to go create a human pathogen.
Yeah.
I mean, this is the thing, and although this kind of work goes behind high biological containment facilities, containment level 3 or even 4, the point about these, and I didn't mention that during my talk, is that no matter how careful you are about the containment facility, it's not 100% foolproof. Incidences of escape of pathogens from high biological containment facilities around the world are very well documented and number in the hundreds. Yeah, they number in the hundreds. Because at the end of the day, it's up to just how, ultimately it's how good your workers are, how rigorous and how careful your workers are, and whether they are really following the rules or not. But even with that, accidental escapes will always happen because no system is perfect. And that is why gain of function research is so dangerous and unnecessary scientifically. That's the key here that we can come back to at the end.
Yeah, I'm sure we'll come back to this in the discussion after the break. I skipped one question, sorry. That's from Paolo Barnard, and he asked for a clarification. Can you address the evident difference in the S2 and S1 binding domain in CoV-2 and its relatives, and how such a large difference cannot be justified by natural evolution?
That's a good question from Paolo there. I think firstly, for me, one is the timeline. There is such a big difference that, and the time span over which SARS-CoV-2 is supposed to have arisen, and when those proximal, these suggested proximal origin viruses existed many years ago, that whether the timeline doesn't really fit, for one thing for me. But also not all of it, and it's not just, we must remember, it's not just the spike protein, the receptor binding domain, that raises question marks. Another aspect of the receptor binding domain that I didn't have time to mention of SARS-CoV-2 is that every single amino acid that is known to be crucial for binding to ACE2 has been preserved, and every other one has been altered. Now that, for me, again, is rather suspicious, as some commentators have raised that, because you expect some to remain, not everyone that is not strictly necessary, although it needs to be conservation of function. Not everyone will be altered, but everyone that is needed has been preserved.
Keep it short, Michael. Keep it short, please.
Yeah, so again, for me, that's suspicious of a lab manipulation rather than a natural selection. There are too many differences, and the types of the quality of the differences as well makes it unlikely that they could have just arisen by mutation and selection.
Thank you. There is one more question, but I think that could be referred to the discussion as well, from Siguna Müller, another ENSSER member. Can you use unique sequences related to the unique furin cleavage sites for better RT-PCR testing? You didn't deal with that, so that's not clarification. And I think we can very well pick that up in the discussion.
Yeah. Yes, I'd like to come back to that, actually. The PCR tests are an important discussion point.
Yeah, and maybe another speaker would like to come back to that too. Thank you very much, Michael. Thank you to the participants for asking for clarification. Let's move on to the second speaker now, That's Professor Ignacio Chapela. Welcome, Ignacio, from the University of California, Berkeley. Michael Antoniou was a molecular geneticist. Ignacio is a microbial ecologist and mycologist. So we're moving into a different field now. And he's talking about the agnotology in COVID. He will explain to you what agnotology means, that it is central to our understanding of science. A historical event orphaned of the scientific regard. That's the title of his talk. Ignacio, the screen is yours.
Thank you very much. This is a sound test. Would you please tell me? Yeah.
Yes, fine.
Thank you. Good morning, good afternoon, good evening. I realize there are people from all parts of the world. It's still dark here. It's the early morning. I have the pleasure and the privilege of being a member of ENSSER, maybe mostly because of my UK and especially my Switzerland connections. But I'm speaking to you from this kind of landscape. These are the mountains of Sonoma and Napa counties, blanketed in smoke as they are, as they were yesterday, reminding us of an unusual situation, a very special historical moment. I cannot forgo just situating myself in this context. And this year, 2020, we will remember here in Northern California for many years to come. But the year 2020 in general, I think, will be remembered for centuries to come, not least because of this COVID situation. What I think could call, why am I not there? What I think I could call the situation COVID. Approaching the situation from the scientific point of view requires, I think, a lot of respect for the situation and also a recognition of the time and spatial proportions of the situation, in that the historical sciences will take a long time, I think, to unwrap this complex situation where we are currently embedded, confused and wrapped in a limited view of what is an incredibly complex context. I would like to consider the scientific regard, not as a way of telling us what to think, but as an approach to thinking about how it is that we think. And the scientific approach, the scientific regard is a very specific one. And here I would like to just use the definition before we get all complicated about it and make sure that the definition is understood that Ian Ball, historian of techniques, provides us, and it's simple: sustained collective critical inquiry. Sustained collective critical inquiry, which is something that I think in ENSSER we really strive to do. We strive within ENSSER to practice a science that is context sensitive, that is time and space sensitive and relevant, that is responsive to the situation and context, that is also responsible. And I don't know why this is jumping. And especially that it is a science practice that is aware of discipline, while at the same time being interdisciplinary in nature. So I will try to make my remarks based on the question of discipline, because I think there is an enormous amount of confusion, a lot of confusion that comes from not being explicit and transparent about what kind of discipline we're talking about and which is the relevant discipline to apply. In opposition to what is presented generally out there as what we should consider to be the scientist's view, I think that we are very aware within ENSSER that what we have is multiple views by multiple scientists. I will have, in the next 20 minutes or so, have three parts, one more general talking about the scientist's views, and then two more in case you feel like you're getting lost or I'm going on too long. The discipline that I would like to utilize for this presentation is a present relatively new discipline, and that is the discipline of agnotology. Born by the coinage of the word in 1992 by Ball and Proctor, it is the systematic inquiry into what is not studied. And even more interestingly, it's the study of how and why ignorance can be induced. A very fruitful application already for a young discipline is an analysis that can allow us to think about the roads that were not taken, the questions that were not asked, the knowledge suppressed, and the promises not kept. You know, the little boy inside me from the 1970s still is waiting for the time when we're all flying around with jetpacks, like I was promised when I was a teenager. And in every field that I have worked in, the promises not kept have always been a bothersome problem, because I'm still waiting now more seriously as an adult for the day when we have anything to say about world hunger that is useful, about disease that makes for better living, and about death that allows us richer lives. So with that agnotological background or frame, at ENSSER, I believe that ENSSER has been working during the COVID period as a forum for agnotological research. We have conducted twice weekly meetings with scientists from across Europe and its peripheries, like California. And as this exception period was announced in late February and early March, and policies were being enacted in our different jurisdictions, our countries, our states, our regions, our localities, each of us had our own opportunity to witness from each of our distinct disciplines and positions the many instances where science was invoked to explain, and it was invoked to justify and to enforce policies of really great significance. Understandably, each of us had many reasons to doubt and even outright reject the process of implementation of this exception period. Some wish to call it the new normal. To the person within ENSSER, members of ENSSER are field tested, so to speak, in making critical analysis count in situations of import to social and environmental questions. And we are also not known for being shy. However, in this situation, in these times of exception, it was clear from the beginning that the situation was quickly developing, not only in the field of virology, but also in other fields, like the field of social engineering and propaganda. The power of political and economic interests, even geopolitical maneuvering, was preconditioning an environment where statements made in the name of science were impossible to consider outside of a polarizing, dividing, and over-determining environment. And so statements made in the name of science, whether they really represented science or not, were and continue to be weaponized. So the weaponization of scientific practice is something that we have been really very aware during this period. So in such a rhetorical environment, and recognizing that we really are set for the longue durée for this situation, we chose to hold back and reflect collectively. So this was the origin of an effort to produce a taxonomy of questions and understand a landscape of inquiry, rather than coming up with answers, with closed answers. And I would like to share with you now, see if I can do this. I need to see some face that tell me, Michael, would you please tell me if you can see an Excel with your head is enough? Just, you don't see? Okay. I'm going to try again. Is that an Excel sheet? Thank you. So this is an extract from what is a living document that cannot be shared publicly yet, because it is being worked on collectively by many different members of ENSSER, where the question is, what questions do we have as individual disciplines from within each one of our disciplines. And I'm not going to go through all these questions. It's simply to mark the diversity of questions that come out from a diversity of scientific approaches and scientific disciplines in an interdisciplinary and responsive environment in which we have been collectively working. So I would like to draw your attention to this column here, where you see different categories of STEM questions, some subordinate questions that have been entered. This is very raw data, and I have extracted a lot of material that that allows me to share this with you without names or anything like that, and no commitments here. Simply to recognize the number of questions, just like the one that that Dr. Antoniou just presented, on the number of questions that are really open to for each one of us. Some of which are really very important, just as important as the origin of the virus. Well, I'll come back to some of these questions, but I just want to give a minute or so of just recognizing the size and the diversity of the question landscape that has been generated through the asking to a series of scientists in collective work. Testing and quantification is an area where I'm particularly interested. But then what's happening with intellectual property rights? Who does the testing? Who owns the testing? And the same thing applies to the vaccine landscape. What's the role of financing and money? Why is it that we don't talk too much about industrialized ag food systems? And even when we talk about any other hypothesis about how this pandemic is developing, we tend to talk about habitat destruction and encroachment of people into wild habitats, but rarely do we hear talk about the role, for example, of pig factories and chicken factories in situations, zoonotics like this. And so on and so forth. The one thing that I would like to highlight from just this quick overview of this landscape of inquiry that has been generated within ENSSER is simply that if there is something we can say, is that there is no consensus. Just like in other fields, in this field that is developing so quickly and so recently, there is really no possibility of anyone claiming to know exactly what the consensus is, what the truth is. This next part, I'm going to talk a little more about what the field where I have something more to say. Even though I don't have enough time, I just want to give you an idea of the map of questions and possibilities that have not been looked at, the agnotological landscape. Before I go there, I really need to make sure that this question is addressed because it generates a huge amount of confusion. It's a confusion that is not often addressed, and in many ways it's exploited by people with all kinds of different interests, and that is the confusion between the virus and the disease. So COVID-19 has this big D to represent or to signal the fact that we're referring to a disease that is associated with a virus. Then the series of numbers and measurements that are generated relating to each one of these different concepts is really a soup of letters and numbers that I, again, don't have time to go through. But for example, just the letter R, that is originally a very simple concept from very simplistic mathematical modeling, has been complicated to generate enormous amounts of confusion because it gets confused with the number R, which is related to the incidence of the virus in a human population in this case, is confused with the functioning of the disease in a human body or in a human population. In the area of testing, it is really important to recognize that there are two very different areas of testing. One is the testing for the virus. The other is testing for the virus within the disease and the response of the human body to that disease. And yet, in the general public and the general media accounts of the situation, these numbers, the numbers that are being thrown at us in the front pages of every news outlet and so on, are really difficult to wade through and certainly confusing. We are living in a situation of massive confusion. And the disciplines that should have counted to look at each one of these situations and each one of these aspects of the situation are being shortchanged as we move forward. I think, as Michael Antoniou just illustrated, in one of these questions, the space for questioning is incredibly large. This is an example here where population ecology and evolution was shortchanged, given an excellent opportunity. This is the Grand Princess cruise ship coming into San Francisco Bay back in March, after having been for more than 10 days going around in circles in the Pacific Ocean. It was allowed to come in and dock at Oakland Airport, Oakland and the Port of Oakland. Let me see if I can do this again. And it was an excellent opportunity, would have been an excellent opportunity to really come up with some of the most important basic numbers that a demographer or an epidemiologist would have liked to produce, because one of these cruise ships is really like it is a vessel where the patients or the human beings were confined, and where many of the basic fundamental epidemiological numbers could have been developed, could have been obtained, had we had the disciplinary curiosity even to find out. There were a few people who tried to get all these people who are here being disgorged from the ship and taken to their different countries. These little tents here are tents where different countries were just collecting their people, putting them in buses and sending them on planes, thereby losing an enormous opportunity very early on in the situation to derive some of the fundamental epidemiological population biology fundamentals that would have been really very helpful. It turns out that just a simple analysis of what is available from that cruise ship and a few others, the numbers that could have been derived then turn out to be months and months, all this time later, and all these millions of dollars later, to have been pretty accurate, even though they were never really calculated properly. Another example, we have missed out on the opportunity to follow Marilyn Rosink's idea of considering the possibility that viruses could be operating in more than the simplistic, simply pathological, pathogenical ecology. In this really good review that I recommend people read, Marilyn goes through many examples and possibilities in which it is shown and demonstrated that viruses are not simply nasty monsters waiting to come and destroy humanity the way we tend to think about the SARS-CoV-2. Viruses are embedded in an ecological and evolutionary context in which we could have been working with the situation completely different had we taken them into account like that. So, for example, we would be having a completely different conversation about what is the value of vaccination, and do we need universal vaccination? Generally, you see a lot of discord in people who think that we shouldn't wait for universal vaccination and move quickly to develop herd immunity. There's a third, and there are many other ways of looking at this situation in which viruses are understood to have this evolutionary attenuation and even incorporation into the mammalian genome. In some cases, there is good evidence that some of this evolutionary attenuation and integration into the mammalian genome provides advantages to populations that become infected with viruses. Is anybody asking these questions? Not quite, not many people. Another missed opportunity or population biology and ecology opportunity is the one that that is more or less well known. I think many of you know about it, and that is the missed opportunity to try and really come up with better numbers about what is the mortality or fatality rates compared to the morbidity rates. That means the difference between the virus moving into a human body or a human population and that virus generating disease, and then that virus generating disease that leads to death. How are those three levels related is still a complete mystery today. And the numbers that are being put around about this subject are completely confusing. It's very difficult to trust any of these numbers, mostly because it would seem as though we really didn't want to know. John Ioannidis at Stanford is someone who has or had at the beginning very good, interesting points of view. Especially, he was one of the people who looked at the Grand Princess cruise ship, trying to come up with this kind of number: fatality rates, morbidity rates, mortality rates, and sadly, he produced his data, and wanting to have them published quickly, he posted them in one of these preprint websites. So he received such an incredible backlash for suggesting that this was not such a deadly virus as it was being said back in February or March, that this paper really hasn't really been seen the day of peer-reviewed light. It's, I think, significant that Ioannidis himself would be someone who is very, very aware of the limitations of published research findings. He was also made famous back in 2005 for publishing this paper on why most published research findings are false. So he's someone who is very aware of the problems of publishing false results or results that might be misleading. Mostly here he approached this from the point of view of statistics. I think Giuseppe Longo, in the following talk, is going to address this from a much deeper perspective mathematically. But Ioannidis here was approaching the question from a statistical perspective. And he says that a research results is less likely to be true, or the statement about a result is likely to be true, when these conditions apply. And here I have time only to highlight two of these conditions where there is greater financial and other interest and prejudice. That means when there is conflict of interest or financial pressure to come up with a specific and univocal answer to a question. And also when more teams are involved in a scientific field in chase of statistical significance, chasing the right answer and hoping to move data around in such a way that you can claim that golden p less than 0.05. For many current scientific fields, claimed research findings may often be simply accurate measures of the prevailing bias, he concludes, Ioannidis concludes. And I think this statement was a prognosis of what was to happen to him, and leads us to the question of how is it that we incorporate the social scientist view into the evaluation of the COVID situation. Again, this is simply an attempt at providing a view, a long view of the space of agnotological possibilities by looking at what disciplines are driving policy and what disciplines are making the story about what happened here in 2020. And if if you look at who is making policy, who is being listened to, and who is developing the story that journalists then reproduce, you see that virology, molecular biology, mathematical modeling, and mechanistic industrialized medicine are really the only disciplines that are taken into account. Of course, some people will go and listen to people like myself who, goes under the flag of molecular of microbial ecology, but these are all disciplines that are left behind. Of course, I need to plug in agnotology here again, but sociology, anthropology, ecological economics, political sciences, evolution, ecology, education, history, psychology, and so on and so forth. Not enough space, not enough time, even to list the disciplines that could have given us a different way of approaching the situation and given us different outcomes of this situation. Who said that what is being now labeled as the new normal was the only possibility? And did we even ask anyone who would know? For example, did we ask educators whether this that is presented as the ideal new normal, very cute way of learning how to write, whether this ever leads to a child learning how to write? Did we ever ask an educator about it? At least I, as a teacher at Berkeley, I was given 3 days to move my teaching from real life to Zoom. There certainly was no consultation. There certainly was no question about it. It was going to happen. And there was no possibility. I, for one, was someone who felt that we could have drawn from historical perspectives and asked the historical sciences about what have people done in similar circumstances. And from the point of view of the historian, I think it's easy to find, without going very far in history, situations in which the response to an emergency was very different. I take, for example, the Second World War situation in London, where children were treated in a very different way. They were moved to residential facilities out in the countryside and taken to school and socializing in those environments. I tried this for my situation at Berkeley, and I offered that I was willing to move into one of our field stations for as long as necessary with a group of students with equipment for the lab and out in the field. And of course, I was told that will not happen. They are beginning to propose the idea of viral bubbles or viral pods, but it shouldn't have taken this long to come to the point where we start thinking about it. Another good example is what happened here in the United States with the CCC camps. Part of the problem, if you asked the proper social scientists, and I'm definitely not pretending to be one of them, is that when the problem is viewed to demand a single unified command, such as the problem of fires, and such as the COVID situation has been presented, as if it was a global fire taking over the planet, more teams are involved in a scientific field in chase of statistical significance. Exactly what Ioannidis said would lead to less reliable statements. And these teams are all chasing a single view to rule the world. So we are left with rule by consensus at best, a presentation as if there was a unified scientific consensus. Secondly, dissent, even questioning an inquiry under these circumstances, must be suppressed at any cost. So we are left with a rule not only by consensus, but a rule by technical expertise, not by science. If I have a certain technical expertise, then I have access to this single view of the world that puts me in the unified command post. Unified commands are used again in massive oil spills, massive fires. Again, this is my local mountainside here. And they require this top-down approach in which one unified commander tells people what to think. And there is no space for thinking about how it is that we're thinking. This definitely does not lead to good science. And I'm afraid it's a situation where we find ourselves today in this COVID situation. The expertise is virology, and we have each country, especially dominant countries in the current geopolitical situation, Germany, France, England, the US, each one of these countries has their favorite virologists that we all know from TV shows. And each country has its favorite modeler, mathematical modelers, people who have very good technical capacity to produce nice models that lend themselves to very pretty and detailed graphs. This is the case of the Ferguson Lab in Imperial College London. That has been, this graph and this model has been ruling our lives for all these months. Whether it was right or wrong, nobody dares to ask anymore. And so we are left, in the best of cases, in an environment under the virologists or the modelers rule, and in the worst of cases, in the hands of spin doctors who are seeking that position of centralized command. I take this as one of the many examples of groups of people who are seeking to be recognized as the ones who carry the word of consensus and who carry the power to suppress. This is the group out of mostly Cornell University, but this is really well documented to be a group that is much too happy to receive money from industry and other interest groups to capture the unified command of meaning making and knowledge making, to claim the mantle of science for whatever it is they decide that is the expertise, the technical expertise that rules the day, whether it be by biotechnology or global warming, or in this case, COVID-19. The question is, who makes or who is the consensus? If when you ask a group of committed scientists, the only answer you really get, or at least the answer that I get, is that consensus is at this moment about COVID really, really far from reality. And who vets the expert? Who is, and who are people who are deciding, who the experts are, who are ruling the moment? These questions are questions that really should be asked and are not being asked. And so we are left on the virologist rule, perhaps, isolated, disorganized, disoriented, hiding behind masks and closed doors, and behind an overwhelming fear of death, waiting for a vaccine that may or may not come, that may or may not be available to each of us, descended as it will be from Olympian heights. Yet a vaccine that we almost take before we are all allowed back into our own lives, orphaned of the regard of science. A new normal, they call it. But I wonder, is this really a new normal that we must accept? And where do we leave science in losing sight of the right perspective? I would say that we really need to claim science back, a science that is open, a science that is inquisitive, a science that is public. Thank you.
Thank you so much, Ignacio. That was very clear as well. There is, I want to go right to the questions. There is a question from Hervé Lemeur. In France, agnotology, he says, designates the science of inducing doubt so as to let industry go on. It was done by tobacco companies, then by climate opponents, now to GMO promoters. See Naomi Oreskes' books and others. It is also the propaganda of Edward Bernays. I would not promote this agnotology. Do you? I think you've been misunderstood if Hervé really thinks you do. Would you say to that, Ignacio?
I think this should also be moved to the questioning, to the discussion section. I seem to recognize that Ian Ball is among us, and maybe he would be the right person to answer that question. But my understanding is that, yes, agnotology was derived from the study of the way doubters, seeders of doubt, the people who plant doubt, how they operate, and the field where this was applied initially was in the tobacco industry. But as the questioner points out very well, the practice of seeding doubt continued to move into different fields. It became a really good practice that was applied to GMO, that was applied to climate change, that is applied now to, that was applied to anti-vaxxers, that now is applied to COVID situations. It's a very well-known formula that is very, very rarely understood to be able to crack it open and see it for what it is. Most people get distracted and fall into the traps that are generated by those people. And agnotology, as I understand it, is the study of that practice in order to understand it and make it visible.
Right, thank you. I don't see any other questions, which would mean that you've been very clear. I think you have, but of course, I've known what you were going to say for a while. But I'm sure that if more questions come up, people will ask them in the discussion, even for clarification. Of course, never hesitate to ask for clarification. Please don't. Just to quote one of my old teachers at my high school, there is no such thing as a stupid question. We have scientist speakers in front of us, so they may use difficult expressions. They're specialists, so if anything is unclear, do ask, even after the break. But now I would like to move on to the third speaker, Professor Giuseppe Longo from CNRS in Paris. Now we move to a mathematician and epistemologist. And you've sensed that the first talk was about hard science, so to say, of the disease that we are struggling with, about the virus in particular. And the second talk was of a different kind. Was about the way you can look at things. That is very much what ENSSER is about, as Ignacio made clear. And Giuseppe is diving deeper into this field. Giuseppe, please, the screen is yours.
Thank you, Dedrix. Can you hear me?
Yes, we can.
Okay, thank you very much. I'm very pleased to be first a member of ENSSER. I'm not a biologist, even though being a mathematician as a background, I've been collaborating with biologists for at least 12 years by now, mostly focusing on new theoretical approach to organisms and its interactions with the ecosystem. I will make more an epistemological reflection than a purely scientific one, though the borderline is very delicate. I think one has to cross it all the time. In particular, I would start with an historical reflection. I would dare to go back to the origin of certain bias on nature that we constructed since the beginning of the scientific revolution. Some 4 centuries ago, we had a fantastic birth of a new science for mathematics and physics, anchored on celestial mechanics, first of all. The care of the stars and the planets was really what changed the perspective in science. But at the same time, there was a major talent in constructing new mechanical devices. And this was due to the real core of the conceptual revolution during mostly the 15th century, first in Italy, then in the 16th century, namely the fantastic mix of observation, theory, and practice, which was totally new, both with respect to the Greek tradition and also Middle Age. The fact that practitioners were theoretizing, like in painting, painters were theoreticians of the perspective, typically in Florence, and the scientists were making observation. Painters were observing nature, like at the highest level, Leonardo da Vinci. But also Galileo was very good in constructing technical devices, machines, and was referring all the time to the artisans in Venice and Florence. However, there was a major bug. These guys, all of them, had a catastrophic view of organisms and life, let's be honest. Descartes and Bacon are probably the best representative, but were far away to be the only ones. The universe is a machine where there is nothing else at all to consider than the figures and the movements of its parts. And Bacon even more. We have to drive nature as a machine through action. Not by chance, it was quoted by a pioneer of biotechnologies, Hartley. who saw agriculture as a source of raw material for industry. This mechanical perspective on physics and mathematics, which was a good start for those disciplines, it was so bad, I believe, for the analysis of life, was clearly understood mathematically by Laplace and Fourier. The nature of determination was clarified at the time. Namely, for these major mathematicians, the mechanicity of the world was captured by what? By the linear decomposition of any reasonable system of equations. So suppose that you can describe a physical process, that we're talking about mathematical physics, of course, and then you have some sort of system of equation. You can always, under certain condition of representability of the physical process, you can always decompose it linearly. This was a crucial assumption that these major mathematicians were doing and did fantastic stuff, like Fourier series, which have been for centuries a fantastic tool. The idea, though, that this was a complete representation of mechanical dynamics, from celestial mechanics to also the ordinary mechanics that happen at our scale. This happened to be shown false within mathematical physics away. By the end of the same century, the 19th century, physics moved away from mechanicism, both as it was intended by Descartes or Galileo, and as it was fully represented by Laplace and Fourier. Why? Because there were negative results. A fundamental way of constructing critical science by showing that something cannot be computed, something cannot be decided, some predictions cannot be made. In particular, Poincaré called negative result his fundamental result, where he showed that the very simple situation of one sun and two planets does not allow the linear approximation of the solution. Almost never one can give, thus, a representation by this cumulative addition of simple components, as it is in the linear decomposition. Because that's the linear decomposition, is the stacking up of elementary simple components to get to the complex. This has been shown false within mathematics. This started a new discipline, which is the geometry of dynamical system, what is called later, a century later, was called complexity theory, chaotic systems, and so on. It started more than 1 century ago. But also quantum physics with indetermination, entanglement, I'm not going into that, departed radically from the Laplacian determination, the mechanistic view transferred into mathematics, and even relativity theory. Because when you understand gravitation and inertia due to continuous deformation of space-time, of course, this is no longer mechanics. Unfortunately, this is not the case for biology. After a beautiful century, the 19th century, with evolution and microbiology, cell theory, physiology, the 20th century started with a strongly mechanical view due to the reading of genetics, the isolation of chromosomes, and then, most of all, after World War II, with the beginning of microbiology. A major text in the theory of these fields, Monod, Hazard, and Nécessité, explicitly refers to Descartes. Not only one place, everywhere. The cell is a Cartesian mechanism, autonomous, exact, independent from external influences. Necessarily stereospecific molecular interaction explain the structure of the code, a Boolean algebra, like in a computer. Of course, now the machine to refer to are no longer the clocks and the gears of the car, is the computer, which has a structural determination which is still of the same type. Namely, you can predict what the isolated computer will do. A fundamental passage, I will go back to this because it's absolutely crucial, is the fact that the stereospecific interaction of macromolecules, namely the fact that the chemical affinities and the geometric structure of macromolecules makes them exactly binding to each other, exactly corresponding, like in the key-lock paradigm, which was at the time revitalized. Some call it simply specificity, but again, this is what was at the core of the idea of the time. It was derived. It is necessary, says Monod, in order to transmit and elaborate genetic information. And then Watson insists, if we could create better human beings through the addition of genes from plants and animals, why deprive ourselves of them? What is the problem? And the surprise is that the genetic specificity is written not with ideograms as in Chinese, but with an alphabet. I told this joke, which unfortunately is not a joke, was not a joke. I told this joke in China, we reached the compromise. Their DNA is written ideograms. Still today, we keep having this terminology, you know, gene editing, typical example of this view. The core idea then is that by the DNA, you can drive the organism in the ecosystem. And indeed, this is at the core of a major researcher in the Human Genome Project. Once they coded the DNA, we will be able to transfer it on a CD-ROM and say, here is a human being, this is me. As Collins, the head of the public consortium for the coding of the human genome, insisted, we have caught the first glimpse of our own instruction book, previously known only to God. Personalized medicine in its normal understanding, in particular as it was understood in the Obama plan, is exactly that. You transfer the DNA of the patient going to the hospital into a DVD, and you send it in the different departments. They will make diagnosis, prognosis, and therapy. Well, eventually, with enough data, with big data and the AI, you will have computers taking care of the cure of the DVD. So you will have machine taking care of the DVD as a coding of the human being. So medicine without patient, without doctor. That's the long-term perspective. I'm not denying the relevance of DNA. DNA is the amazing physico-chemical trace of evolution. Yet we have to move away from the genocentric perspective of DNA as driving the organism in the ecosystem, which is the core issue today. I would like to insist about the critique of the core principles of genocentrism, the unidirectional exact transmission of information, and then get to the point how this is of interest to us. The exact stereospecificity of macromolecular correspondence, which was an extension to macromolecular interactions of an ancient reference to the chemistry of the 19th century, and the central dogma of molecular biology. Still now, you find this perspective present in major references, because these are the gears of life, macromolecules, so they act like gears in Cartesian mechanism. That's what against evidence has been kept saying against evidence, because we know very well something which is beautifully summarized now as even more radically, proteins never do fold in a particular shape, but rather remain unstructured or disordered. In mammals, about 75% of signaling proteins and half of all proteins are thought to contain long, disordered regions, while about 25% of all proteins are predicted to be fully disordered. Many of these intrinsically unstructured proteins are involved in regulatory processes and are often at the center of large protein interaction networks. That's a very different situation we have to face, which of course leads to stability, but in a quite different way from Cartesian mechanism. Unfortunately, these principles, the exact specificity of interactions and the central dogma are still taught in textbook. You find it in all medicine textbook for first, second year students, and very often also in biology. It's no longer as it was phrased in 1960, namely the unidirectional transmission of information from the gene to DNA to RNA to the proteins. It is represented in a slightly more complex way, but it is still there. In research, not many refer explicitly any longer. It is suddenly starting to disappear, but implicit is everywhere. In particular, it is in the practice of research in GMOs and other fields I will mention, and also in virology. But what is a gene in this context? Evelyn Fox-Seller nicely points out that this notion has been changing 5 times at least in the 20th century. I would like to quote, as for this, a major personality in gene research, Francis Collins, who was head of the Human Genome Project. In 1999, in a paper, he writes that we expect to find 80,000 genes. This is the consequence of a perspective he had been developing for about 20 years, namely the idea that there is one gene, one protein, or well, one gene, one enzyme, which was the result of Badel and Tatum work in 1940-41, and the result in a Nobel Prize. That's a typical case of a differential method. I mean, the experimental insight by Badel and Tatum was a major one, no doubt. I mean, it was fantastic to achieve this kind of results, or analyzing and using mutation in the chromosomes and then observing well before 53, observing changes in an enzyme. But of course, it doesn't give per se a correspondence, a positive correspondence. A difference inducing a difference in a complex network of interaction doesn't prove a direct connection. Yet still, Collins believed this, that's why he derived his conjecture, 80,000 genes. 2 years later, he himself said, oh no, we have 25,000 genes. 3 years later, well, perhaps there are 20,000. What is the situation? Well, first of all, since 2002, there's an increase in literature about the stochasticity of gene expression. There is alternative splicing, too complex now to explain. There's overlapping genes. That's something which is increasing importance, in particular in viruses. And I will go back to this. What does it mean? Here there is an image. I mean, with some binding, you define D, C, and A as genes, but they can express if they were A, K, and B according to the context. The context may modify the overlapping of different genes that were supposed to be distinct and producing, expressing differently, which is false. This was known since long, and as many other alternative paths were hidden for too long, and now is exploding, in particular in presence of this virus. Indeed, that's what I would like to go to in this perspective. The coronavirus that I know was decoded very early in January, which is, of course, fantastic knowledge. We have seen how this was used by Michael before, because you understand the evolutionary path, and that's crucial. I mean, in theory of evolution, the decoding of DNA of animals is enriching enormously a beautiful theory. In spite of the various schools, the beautiful theoretical debate in theory of evolution in 200 years, about 200 years, Lamarck, Buffon, and most of all Darwin, has allowed to embed the knowledge of DNA in a theoretical apparatus that justifies it, and so let's understand histories, and that's fantastic. But then for the understanding of the virus behavior, how does it behave in a context? What do you know from the decoding? Symptoms can range from nil to mild to severe, to having to go to hospital, to being on life support, to death. Some people recover quickly, others make many months of rehabilitation to recover. Some never seem to fully recover. For example, lungs may be so scared that they may never fully recover. There are also neural damages. There's increasing weakness. By the way, SARS-CoV-2 seems to contain a high number of overlapping genes. That is, the context drives the expression of the RNA, in this case, the interaction in the context. So the question, is this a zoonosis? Well, I read a beautiful book by Moran and Figuier, and with a beautiful historical record of 70 years or 60 years of epidemics. The first remarks in this direction date from 93. And in that book of 2015, so well before the beginning of COVID-19 shows many graphs and a lot of evidence that I summarize here from this graph from their book, where it is shown the amazing increase of epidemics. Some of them may get to be pandemics, more rarely. Epidemics stay local. The black ones are those that recur, that come back about every year. The others are singular events. The point is that there is massive evidence that about 70% of them are zoonoses. This is largely due to the interfaces with the ecosystem that we handle like a machine, that we think we can intervene on the ecosystem and demolish, destroy, and act on trees, on animals, on species, as if they were resources, as it was theoretized, as I have called before, since the early time of biotechnologies. So by this use of the interface with animals, wild animals, of course, is a good reason for transferring of viruses to animals in bred and then sometimes to humans. Well, we heard a beautiful discussion by Michael whether the virus has been artificially edited. I have no idea. I entirely trust his views, and I think his arguments were very robust. But I would add my epistemological consideration. We are in a situation where lots of disciplines converge to give fantastic technologies coming from chemistry, crystallography, electronics, informatics, computer science has a major role, in the hands of genocentric fanatics, because that's the point. I developed in a paper an analogy with the geocentric perspective. You see, it is as if we had given to people with geocentric perspective, Ptolemaic perspective, the talent, the technique, the technologies to construct a rocket capable to reach Mars. They would never get to Mars because, of course, by looking at the sky with epicycles, they would never get there. But even worse, as they would not be able to consider the speed of rotation of the Earth, their rocket would fall on the next city. We are exactly in this situation. So that's why I insist that we should have, first of all, before any other social, ethical, ecosystemic precaution, a principle of scientific precaution. Since GMOs, gene editing, gene driving are grounded on the central dogma and key lock metaphor, or are handled with those perspectives, even though sometimes now are no longer made explicit, but that's their background. GMOs are the direct children of the central dogma. And the other key issue that I mentioned before, exact stereospecificity, we should simply say no, because this is we have powerful technology in the hands of a wrong global scientific perspective. I insist again, I don't deny the major relevance of DNA, of course, but we have to see it from the point of view of the organism and the ecosystem, because the DNA is continually used by the cell in a context. I would quote here a very interesting case, the case of the fish, cave fish. There are more than 100 species, close species, of fishes that have lost their eyes, living in caves for many million years. What's very interesting is that most hybrid offsprings that have been produced have eyes and have functional eyes. So the DNA is there. The context, evolution have been driving the use of DNA in different ways according to the context. So molecular biology gave us fantastic data and mechanisms. Again, the analogy can be made with the fantastic Ptolemaic astronomy, in particular in the Arab world. The astronomers between the 7th and the 12th century, the 13th century in the Arabic world knew all possible positions and relative movements of all visible stars and planets. They knew all mechanisms. They could say when Mars is in that constellation, then Jupiter is close to the other one, or when Venus is there, there will be certainly Saturnus there. So they knew all the mechanisms, and they could establish even causal relations, which of course were all wrong, because the theoretical frame was wrong. The mechanisms where the data were very useful. Consider that the Alphonsine table, which were produced in the Catholic Spain on the grounds of the Arabic production of data and mechanisms, were the tool for navigation. So America was discovered using those data and mechanisms. The theoretical frame, totally wrong. The causal relation, totally wrong. We are in a similar situation. So I think we should move toward new principle. Many people, including my colleagues in the States, work in a different perspective. We consider that macromolecular interactions are largely stochastic. They must be given in probabilities, and these probabilities depend on the context. We even dare to say that almost all most common phenotypes use almost all DNA, namely in each cell, each fragment of DNA is used or expressed, perhaps with the role of control. This fact is that the cell, the organism, and the ecosystem strongly canalize, constrain, and select macromolecular interactions, starting with gene expression. So we are moving from a vision of organisms that way, which allowed the myth of driving animal, plants, and viruses in the ecosystem, like a machine, to a very different perspective that is the more holistic perspective based on complex interactions with all levels of organizations. Let me quote some books that I mentioned here. I strongly recommend The Music of Life in particular. Unfortunately, I think that the book by Kupiec and Sonigo is not in English yet. And I would like also to recommend a major paper in the direction I mentioned before for a more organicistic perspective. Okay, I think I would be pleased to interact now and stay by this in the time that was given and go again to the discussion.
Right, thank you very much. Ignacio, sorry, Giuseppe. You will have noticed, I'm speaking to the audience now, you will have noticed that only the first of these three talks is about the scientific matter, the life sciences of COVID-19, and the second and the third ones are about scientific knowledge as such. We really want, ENSSER really wants to help you think for yourself, which is a hard thing to do. But that's what we want to say. That's one of the things we want to say with the second and the third talk. There is one question in the Q&A box for Giuseppe. That's from Siguna Müller. She says, reversing or stepping away from the unidirectionality of the central dogma and from seeing viruses as a machine, what if for viruses, metabolism is one of the key drivers, perhaps sometimes more influential than the genome? If you have a short answer, Giuseppe, yes, but this is really a typical question for the discussion. Again, it's a very good one. Thank you very much, Siguna.
I think that since 1961, from Durant work, we know that metabolism may modify not only gene expression in plants, but in plants, even the DNA structure. So, of course, this is highly possible. And it was one of the matters that was denied by the central dogma, while we know that metabolism has a major role, even, for example, without modifying DNA, but surely in gene expression. Well, of course, the bees and the ants are good examples known since 10,000 years, I would say. Yeah, that's a key issue.
Thank you very much. I don't see any other questions right now. As I said before, please do not hesitate to ask clarification questions in the discussion after the break as well. You should never be left with any uncertainty as to what the speakers have actually said. That's essential. But I would now like to hand over to the moderator you'll see after the break, Dr. Angelika Hilbeck. Thank you very much to the speakers so far. They will appear again after the break, and Angelika will lead you into the break before we have the break.
Exactly, that shall happen. Hello, everybody from my side. Good to see you or not see you, and hope to see you all after the break. And I will kick off then the debate among the participants or among the presenters. And then hopefully pretty quickly after that, we will also engage you in the debate, and you will be able to make oral statements or to post oral questions there as well. We kept it right now to the written part when it was about clarifications. The next one is now really about engaging in a conversation, also in a conversation with you. And we believe that even though we cannot be in the same room as persons and as people, we do want to hear your voice, but we cannot unfortunately see your camera. That the technology doesn't allow us to do that, but we do want you in the room by your voice. So I look forward to it. Take a break, have a coffee, but do come back, because I think some of the more interesting or really interesting questions that might not have been addressed now will come during the roundtable discussion. So I'll see you in, Friedrich.
Half an hour at 4 o'clock exactly.
4 o'clock.
4 o'clock Central European time, that is. I hope you can calculate what it is for you.
You have to do the math on your own. All right.